Have you ever really thought about what's going on in a forgotten container in your fridge, or what makes those rocks in a stream so slippery? Well, we're surrounded by a totally invisible world of microbial growth, and this process, it dictates everything from our health to our food. So let's pull back the curtain on this hidden universe, and we're going to start with something I guarantee you've seen before.
And yeah, I mean that literally. That slime isn't just some random gunk. It is a bustling, highly organized city of microorganisms. It's a full-blown ecosystem with its own infrastructure, its own communication networks, and even defense systems.
The official name for this is a biofilm, and you can think of it like a fortress that the microbes build for themselves. They secrete this gooey substance, this matrix that holds everything together. And this structure, it protects them from all sorts of things: harsh conditions, our immune systems, and even antibiotics.
And they are absolutely everywhere. You know that plaque your dentist is always scraping off your teeth? That's a biofilm. The gunk that can build up inside a medical catheter, that's a biofilm too. They thrive out in nature, but they're also a massive challenge in medicine, causing these really persistent, hard-to-treat infections.
So, how does a single microbe land on a surface and build one of these incredible cities? Well, it all comes down to a remarkably simple and efficient engine of growth. So let's zoom in from the scale of the city right down to the individual citizen.
The secret is a process called binary fission. And honestly, it is elegantly simple and just brutally effective. One cell becomes two, those two become four, then 8, 16. You get the idea. It's the simple, powerful engine that drives their explosive growth.
And what's amazing is just how organized this whole thing is. First, the cell bulks up a bit and makes a copy of its DNA. Then, this special protein called FTZZ forms a perfect ring right in the middle. It acts like a little contractor, building a dividing wall or a septum. And once that's in place, the cell just pinches into two. It's simple, it's fast, and it is absolutely perfect for exponential growth. So, how fast are we talking about? Let's take a single E. coli bacterium. Under perfect conditions, it can double every 20 minutes. If you just let that one cell divide for 24 hours, you'd end up with this many cells. That's over 281 trillion. Just take a second to look at that number. It's a scale that is almost impossible to wrap your head around, and it's exactly why a tiny contamination can become a huge problem literally overnight.
But here's the thing, a population can't grow like that forever. In a closed system, like a lab culture or a sealed container of food, a bacterial colony goes through this really predictable life cycle. And you can think of it like a 4-act play.
Act 1 is the lag phase. So, the bacteria have just shown up in a new place. They're not dividing yet. Instead, they're kind of getting acclimated, repairing any damage, and making all the new proteins they need to get going. They're gearing up for what's next. Then comes Act 2, the log phase. This is the main event. The population just explodes, doubling at this constant, super fast rate. And here's why this is so important to know. This is when the bacteria are most active, and that also makes them the most vulnerable to antibiotics that work by targeting things like cell wall construction.
In Act 3, things start to slow down. This is the stationary phase, and the party's kind of winding down. Nutrients are getting scarce, and toxic waste products are starting to build up. So the growth rate stalls. The number of new cells being made is about equal to the number of cells dying off. It's a stalemate.
And finally, we have Act 4, the death phase. The environment has just become too toxic, and there aren't enough resources left. So cells start dying off way faster than they can divide, and the whole population crashes. And that's the end of the play. So, what is it that determines how fast bacteria move through this cycle? Well, it turns out they're a lot like Goldilocks. For them to really thrive, their environment can't be too hot or too cold, too acidic or too basic. Everything has to be just right. Let's start with a big one. We all tend to think of oxygen as being totally essential for life, right? But is that actually true for everything? For a whole lot of microbes, the answer is a resounding no. You have these two fascinating extremes. On one side, you've got the obligate aerobes, like the bacteria that cause tuberculosis, which absolutely must have oxygen to survive. But then on the other side, you have obligate anaerobes, like the bacteria that cause tetanus. For them, oxygen is literally poison. It kills them.
But of course, it gets way more nuanced than that. Between those two extremes, you've got a whole spectrum. You have facultative anaerobes, like E. coli, which are super adaptable. They'd prefer to use oxygen, but if it's not around, they can switch gears. Aerotolerant anaerobes, they just don't care. They totally ignore oxygen. And then you have the microaerophiles, the true Goldilocks of the bunch. They need oxygen, but only in small, just right amounts.
And you see the exact same principle with pH. Most bacteria, especially the ones that make us sick, are neutrophils. They like a nice neutral pH around 7, which is, just like our bodies. But then you have acidophils that thrive in super acidic places like a sulfur mud field, and alkalophiles that prefer extremely basic conditions like you'd find at a soda lake. And then, of course, there's temperature. Psychrophiles, they love the cold. Thermophiles, as you can probably guess, love the heat. Think hot springs. But the group that's most important for us to know about are the mesophiles, the middle-loving microbes. And there's a really simple reason why. Their perfect temperature range is right around our own body temperature, which makes them perfectly adapted to cause infections in us. Okay, so why does all of this matter? Well, it's pretty simple. Once you understand the rules that govern this unseen world, you can learn how to control it. And this knowledge is the basis for so many technologies we use every day. When you put food in the fridge, you're actually just slowing down the growth of those mesophiles. Pickling food uses high acidity to do the same thing. Doctors prescribe antibiotics specifically to hit bacteria when they're in that vulnerable log phase. And those autoclaves in hospitals, they use extreme heat to create an environment where nothing, not even the toughest heat lovers, can possibly survive. So we've seen how these tiny organisms build actual cities, reproduce at these insane rates, and live by a really strict set of environmental rules. So the real question is, now that you understand their rules, how does that change the way you see the world around you? Because the next time you salt your food or wash your hands with soap, you're not just cooking or cleaning, you're actively manipulating an entire unseen ecosystem.